I'm going to be telling you a little glimpse of what we know about influenza virus, how it causes epidemics, how it causes pandemics, some of the aspects that we have also been studying in my lab, and finally finish with what are our thoughts with respect to the possibility of a universal influenza virus vaccine, and what are we doing in this respect with the possibility of developing such a vaccine. And I want to thank NIAID, that's the National Institutes of Health, for funding in the form of a Center of Excellence in Influenza Research and Surveillance. There are 5 of such centers funded by NIH in the United States, and our center, we call it CREEP, which is Center for Research on Influenza Pathogenesis. And many of the studies that you have heard about dual-use research concern and things like that, unfortunately, they have been done by people that have been in my center as subcontractors. We can spend a lot of time talking about these issues, and I would be happy to talk about it after the talk with anybody that wants to know. But I'm not going to talk. I'm not going to touch the dual-use research concerns at this time. So, influenza virus. Influenza virus is an RNA virus that contains, and you can see here in this electron microscopy that was made first by Yoshi Kawaoka group, and now it has been seen by many different groups. When you do a nice electron micrograph of the virus, you can see, in addition to the envelope, you can see the RNA segments, and you see 8 dots corresponding each to one of the RNA segments of the influenza virus. Each RNA segment is going to encode 1 or 2 proteins. And in the envelope, you have these spikes that are representing the hemagglutinin and the neuraminidase of the virus. These are proteins that are important for entry and for exit of the virus, and they are the basis of the nomenclature of the influenza virus, with H1, M1, H, the 1 corresponding to antigenic group 1 of the hemagglutinin, and the 1 here in the N, antigenic group 1 of the neuraminidase. And this is now a cartoon representing the same virus with the 8 different segments, each one encoding 1 or 2 proteins. And in the envelope again, the hemagglutinin and the neuraminidase. Hemagglutinin is the one that binds to the receptor, and neuraminidase is the target of the only now approved antivirals against influenza that can be used against influenza. They inhibit the neuraminidase activity of this protein, and because this protein is very important for the virus to spread, then the neuraminidase inhibitors are going to inhibit virus spread, and that's the basis for how they are working. The polymerase proteins, they are encoded by 3 genes, PB2, PB1, and PA, and there is a lot of interest now in finding inhibitors of these proteins, especially because now the crystal structure of these proteins has been recently published by the group of George Cusack, and there is a lot of hope that one can get now, again, inhibitors against these proteins that will complement or will enrich the type of inhibitors that we have against influenza. The virus also encodes a nucleoprotein that covers all the RNAs in the form of a viral ribonucleoprotein, and this is the infectious RNA of the virus, which contains both the RNA, the nucleoprotein, and the polymerase. Without the polymerase, the RNA is not infectious because it needs to be transcribed first by the polymerase in order to be infectious. Another proteins that are encoded by the virus, the matrix protein, which is underneath the lipid bilayer, and also the M2 encoded by the same gene, which is a transmembrane protein that is important for viral entry. Ion channel activity is necessary for proper viral entry of this protein. And then there is the NS protein that encodes the NS1, the non-structural protein inhibiting interferon, as well as the nuclear spore protein, which is involved in trafficking of the RNPs. Now, in terms of influenza virus in humans, we have 2 types of influenza viruses, type A and type B. And these are co-circulating in humans and have been co-circulating in humans since we know of, especially since we have isolated influenza virus, and then we know that influenza is caused by influenza viruses. And both influenza type B and influenza type A, they are able to reinfect people that have been infected with influenza virus. And that's unusual for an RNA virus. So you get infected with measles, for example, which is another RNA virus. People that get infected now develop long-lasting immunity that will prevent reinfection with measles. So why this doesn't happen with influenza virus? Actually, this happened with influenza virus. If a person gets infected, for example, in 1980 with the influenza B virus that was circulating in the 1980, it develops long-lasting immunity against this strain of influenza B virus that was circulating in 1980. The same thing happened with influenza A virus. The problem is that the virus that comes later on in the 2000s is different from the virus that infected in 1980. And it has antigenically changed enough in order to be able to evade pre-existing immunity. So these viruses, influenza B and influenza A, they change year to year slightly, and by these changes they are evading pre-existing immunity in people that was reinfected previously with influenza. After 10 years, these slight changes start to be more dramatic changes. So in general, someone that has been infected with a B virus becomes possible to be reinfected 10 years later. So this is what is called antigenic drift. The virus drifts antigenically from year to year. But in influenza A, something even more dramatic can happen, which is what we call antigenic shift. There is a big antigenic change, not just a slightly antigenic change from year to year, which is what is called shift. And this results in these changes from H1 to H2 to H3, etc. So again, the 1 refers to an antigenic type 1 for the hemagglutinin, and since 1918 we have had in humans H1N1s in 1957, in which they dramatically disappeared and they were replaced with a completely different hemagglutinin containing virus, antigenically not related or very poorly related to H1, to the point that antibodies against this H1 are unable to neutralize at all these antibodies against H2. And when this happens is when pandemics happen. And pandemics have happened in 1918 with the introduction of H1s, 1957 with introductions of H2 in humans, 1968 with introductions of H3 in humans. Every time that the new subtype came, the previous subtype disappeared from the human population. Now in 1977, H1N1s came back to the human population, and the H3 and 2s did not disappear. The difference between these introductions here and this one is that this virus that came in 1977 was almost identical to the virus that disappeared in the 1950s, to the point that it's believed that actually it came most likely from a laboratory strain that was released into people. And I can again talk about what are the speculations after my talk, if someone wants to know. What are the speculations about how this virus was released again from a laboratory? But this virus didn't cause a pandemic, just started to go circulating with H3s. And then in 2009 there came another pandemic virus. This is an H1 that now, yes, it was dramatically different from other H1s, to the point that there was very little antigenicity that was related between these H1s and the H1s that were circulating, and because of that there was no pre-existing immunity again in humans against this H1, and this resulted now in the pandemics of 2009. This virus made this all H1 to disappear, but the H3s didn't disappear. So today we have in humans influenza B virus, influenza H1 and influenza H3. B virus, there are actually two different lineages. Sometimes one year is more, or one lineage, some year is more of another lineage. And for this reason, most of the vaccines against influenza, they are now tetravalent. They have two B components, one H1 component and one H3 component. So 4 different antigens in the influenza virus vaccine. But not only that, the vaccine needs to be updated almost every year. And that's again because the virus changes from year to year. And we know, for example, this year, the vaccine, that H3 component that was included in the vaccine doesn't appear to match antigenically the H3 viruses that are circulating, and because of that there is a lot of activity with H3, even in people that is vaccinated, and we are expecting to have a very bad H3 year this year. Now, there is more subtypes of influenza virus, and we get reminded of these subtypes because from time to time there is people that get infected with these subtypes. The difference between viruses that have started the pandemic and these other ones is that although some people get infected, there is no good propagation from person to person. This has happened with H5N1s, H9N2s, H7N7, swine H3N2s, H7N9s, and much others that are not included here. The ones that normally we are more concerned about, that because they are also more abundant in terms of human infections, are the H5N1s, the H7N9s, and these swine H3N2 variants. And we still don't know which is going to be the next pandemic. In 2009, we could not predict, we have not predicted that the pandemic will have been caused by a new H1N1. Not only that, this virus was completely unknown. It came, look it like almost from nowhere. We know it came from swine, but it has not been detected in swine by our surveillance methodology before it jumped into humans. So it's very difficult to predict what is going to be the next pandemic, and the vaccines against the currently circulating viruses are unable to prevent infections when these changes in antigenicity happen. Now, where these different subtypes come from? They come from birds. So we have as much as 16 different hemagglutinin antigenic subtypes circulating in birds, and 9 different denominated subtypes circulating in birds. And they are in birds, in wild birds. It doesn't cause a very disease as much as we know. They are well adapted to these animals. And they cause intestinal infections, and they are propagated by the fecal oral route. But from time to time, some of these strains jump into different species, and they may establish also their own adapted strains. So we have strains also that are circulating in poultry, different from the ones that are circulating in wild birds, strains circulating in pigs, strains circulating in humans, strains circulating in horses, and more recently, strains circulating in dogs. These strains usually are adapted to the host, and they stay in the host. But sometimes one of these strains jump from one host to another, and then somehow adapt to this host, starts to propagate in this host, and then results in the introduction of a new subtype of influenza virus in this particular host. When this happens into humans, then you get a pandemic. And the hemagglutinin subtypes that have come into humans, they have come either from poultry, when pandemics occur, or from swine influenza. Now I'm going to start talking a little bit about the first pandemic virus that we know of, which is 1918 H1N1 influenza virus. And this is work that started in my laboratory by a postdoc, Alicia Solórzano. We have done it in collaboration with Peter Palesa and Chris Basler, the CDC, the USDA, and the Armed Forces Institute of Pathology, Jeff Taubenberger. And we became interested in 1918 because of the high virulence that has been associated with this particular strain of influenza virus in humans, which has been unprecedented and has not been seen again. And one of the best ways how to illustrate that is looking to, in this case, the US life expectancy, and then you see in the last century, and then you see it increasing because of the modern advances of medicine, the hygienic techniques, hospitals, modern medicine, etc. Except for this big dip that was here in 1918, and this big dip was caused by the influenza virus. So there was a lot of people that died of influenza virus infection in 1918, and because of that, they make a dramatic go down in the life expectancy in the US and all over the world. So there was something peculiar about this particular infection. Here is another way how to illustrate the impact of 1918. This is a small village in Alaska after they had the 1918 influenza virus infection. So they were, the infection was spread in these remote areas, most likely through the mail, the postman that comes with flu, infect the people, and then in these remote areas, which probably they were less exposed to influenza, the infection was even more traumatic. Almost every adult died. So the only survivors that you see here are kids. All the adult human population was wiped out from this particular village. That was not so dramatic in non-remote areas, but still there was a lot of normally healthy adults that died in 1918 of influenza virus infection. And why was that? What was so peculiar about this virus? Now, the problem about studying this virus is that this virus didn't exist at the time that we were able to study influenza viruses. This virus was never isolated. It was not even known in 1918 that influenza was caused by a virus. And what made possible to study this virus was two people, mainly. One is Dr. Helting, which in 1997 came to a small village, again in Alaska, a big mission, that most of the adult population, like I saw in this village, or also in the previous picture, died of influenza virus infection in 1918. And then the army came there to this village to bury the people, and they buried the people in a common falls under the permafrost line. So that means, it was north in Alaska, that means that these bodies have been frozen since 1918. This Dr. Helting came there to the mission, exhumated the tombs there, and then picked up samples that they were not very well preserved, but some samples, tissue samples from people that died there in 1918. There is no infectious virus in these samples, but what there is in there is still pieces of the RNA of the virus that has been degraded, but the pieces are there. And the other source where one can find pieces of RNA of people that died in 1918 of influenza virus infections are these paraffin-embedded blocks, which are coming from lung samples of soldiers that died in 1918, American soldiers, died in 1918 of influenza virus infection, and they were preserved under a old biobank preservation act from the United States government. They were preserved in freezers with the hope to study later on what could have been caused this particular disease. And again, the virus is not there, but there are pieces of RNA. And by extracting RNA from these samples and using now modern sequencing techniques, amplification and sequencing techniques, has been possible by Jeff Taubenberger to sequence now the genome of the 1918 virus and now clone the genes that encode the virus. And we started a collaboration with Jeff Taubenberger. At this moment, we have developed techniques to make influenza viruses from recombinant DNA. So you can take a specimen of person that died of influenza virus infection in 1918, sequence the genes, reconstruct the genes now using DNA synthesis, and using reverse genetics from plasmid DNA, recover infectious viruses corresponding to the genome of the 1918 virus, and then now study this virus in tissue culture animal models. Something peculiar about this virus that makes it more virulent. And obviously we do that under high biocontainment, and we do that in collaboration. We have been doing that in collaboration with the CDC in the United States. So that's how the virus looks like. It looks like almost any other influenza virus. There is no apparent morphology that makes this virus peculiar. There is not even apparent sequence that will have made this virus alarming, like any known motif in the sequence that is associated with high pathogenicity, like some of the motifs that are present in H5N1s, which relate to hemagglutinin, the presence of a multi-basic cleavage site in a portion of the hemagglutinin. This makes these H5 viruses highly pathogenic. This sequence was not there in the 1918 virus, so it was not apparent from the sequence that this virus was going to be virulent. And when you put in animal models, then things change. So you take and inoculate mice with 10 to the 6 PFU of a H1N1 virus that was circulating in humans in 1991. The animals look quite happy, they don't lose weight, and they survive infection. But if you include 5 genes of 1918 and now 3 genes, all the genes except the polymerase genes of this Texas 91, now the animals are succumbing to death and they die, the mice, in a few days. And if you even replace the polymerase genes, these 3 polymerase genes by the 1918, so this is the complete 1918, the animals die even faster. Now, if you just replace the hemagglutinin of 1918 with the hemagglutinin of Texas 91, then now the animals again don't die. So this experiment is telling us that virulence, at least in mice, is multigenic. you require the hemagglutinin, you require also the polymerase genes for fully virulence. And if you look to viral titers at day 4, what you can see is that at least in this system and using this virus that we are using, virulence correlates with the ability of the virus to replicate to high titers. So the Texas 91 that does not kill mice grows to 10 to the 3 PFU. This one that kills mice, 10 to the 6. The complete 1918 grows to 10 to the 8. And you replace the hemagglutinin and goes back to 10 to the 5. So growth in mice to 10 to the 5 or less, survival, more than 10 to the 5, then death, and the more viruses, the most likely you get death. This is now an experiment in ferrets. This is looking to the lungs of ferrets infected with, again, an H1N1, 1991. What you can see is there is almost no sign of infection, no sign of inflammation. The lungs look quite happy. If you take the same ferret and infect it with 1918, you get a completely different picture. The virus, the lungs are completely inflamed. These animals die of pneumonia. So what do we know now about this virus? After we studied them, we have studied in different animal models, both my group and the group of Yoshi Kawaoka that also reconstructed independently in Canada. And it's the only known human influenza virus that is lethal in several systems: mice, ferrets, macaques, and embryonic eggs. So this is telling us that this virus really was a quite virulent virus. Maybe there was some predisposition conditions in 1918 that make influenza virus more virulent than usual in people, but also this one we cannot exclude, but what we can say is that at least this virus was quite virulent as compared to other H1N1 viruses that were secreted in humans later on. We know that the virulence is multigenic, so it's like a perfect storm. You require several genes that encodes pathogenicity. The glycoproteins are essential for the virulence, HA and NA, and also the non-structural proteins. I have not shown that, but it's also essential, as well as a gene that is in the polymerase gene that is also non-structural, PE1F2. But we also learned some positive things. We reconstructed the virus. We looked whether it was sensitive to existing antivirals, and it is. So even if we will get now infected with a virus like that, at least we can try to treat people with antivirals. And we know that the vaccines that are based in a homologous strain are protective too. So at least we know that now we can treat and we can protect also people against a virus like 1918, if a virus like 1918 occurs again in the human population. So if we ask the question, would the 1918 like H1N1 virus be today as lethal as in 1918? Probably not, because now if we have time, which is one of the problems, but if we have time to vaccinate people, then it will not be as lethal. And we have also antivirals that can help in the treatment of infection. With that, I go now to when H1N1 caused again a pandemic. That was 2009. A very different pandemic from the pandemic of 1918. It didn't cause so much lethal disease. It was actually the mildest of the pandemics that occur for influenza virus pandemics. And I'm going to show you now data from two postdocs in my lab, Raf and Balaji Manikasamy, in collaboration with another postdoc in the lab of Peter Palese, and we have been collaborating also with different people, including again the CDC and Bob Belsi for human studies. So again, we have here this virus, this pandemic H1N1 that came in 2009, and where this virus came from, why this virus caused a pandemic. And this came from swine. So it is what is called swine flu. It's a virus, the hemagglutinin didn't come from an avian reservoir, which is what happened in the H3s and H2 pandemics. In this case, it came from a swine reservoir. And what we know about the origin of this virus is that in swine, there have been circulating different lineages of influenza viruses. One that is called a classical lineage, here in pink, and the other is a Eurasian lineage. This was in North America. This was in Eurasia. Somehow these two viruses met, it's still unclear where. Probably in a pig, but where in the world is unclear, because in general the North American viruses don't mix with the Eurasian viruses for pigs. And not only these two viruses mix, but they also mix with a human H3N2 and an avian virus and created this, what is called quadruple reassortant. different genes from different species, but this virus has been circulating in pigs for now many years before jumping into humans. So even this quadruple virus could be considered a pig virus. And the important thing is the hemagglutinin, where the hemagglutinin came from. The hemagglutinin came from the classical swine influenza viruses, H1N1. And one thing that we learned about this one is the 1918 PB1F2 was not present in this virus. So despite that it was, again, in H1N1, just by looking to the sequence, what we have predicted that it will have not been as virulent as the 1918 because it lacks one of the most important virulence factors for 1918. Now the hemagglutinin in pigs came actually, the ancestor is again the 1918 virus. So the 1918 virus, the H1N1s, when they came, they were very virulent in humans, but they started drifted, and they have been drifted in humans till the 2009 pandemic came to form the modern human H1N1s before 2009. And this hemagglutinin now is very different from the hemagglutinin of 1918 because it has been evolving and adaptive to immune response in humans. But 1918 went also into pigs and caused also disease in pigs and caused a lineage in pigs, which is the classical swine influenza virus. And in pigs, the hemagglutinin didn't change so much, probably because there is less antigenic pressure. And why there is less antigenic pressure is because pigs are short-lived in general, so they can be infected with flu because of the short life of pigs, most likely only once in their life. So flu does not need to evade persistent immunity. And that might be one of the reasons why this virus has a hemagglutinin that was antigenically more similar to the 1980. But this allows us to make this particular hypothesis. When the pandemic H1N1 came, the infections were primarily seen in children and young adults, and not in the elderly. And the elderly usually are the people that are more susceptible to severe disease by influenza virus. So that was actually good news because the virus was not circulating well in the elderly, in the old people, and because of that was also not causing severe disease because most of the severe disease is there in the old people. And funny enough, the first serology studies that were done to look for antibodies against this pandemic H1N1 in people so that there is some cross-protection in antibodies found in sera of people that are older than 65 years old. So were these antibodies in these people, in the old people, where did they come from? And they were probably the ones that they were protecting the old people against severe infection with the new H1N1. So we already postulated that it could have been done to prior exposure to an H1 virus similar to a 2009 H1N1. So again, viruses that were circulating here in humans during this time antigenically may be more similar to the HA from the swine influenza virus, and antibodies developing in humans exposed to these strains that are still present there after so many years, they could be still protecting against infection with the classical swine flu or the pandemic H1N1. So in order to address this question, do antibodies to any specific H1N1 virus protect against this virus? We generate antibodies against different strains of influenza virus, the 2009 pandemic one, but also strains from the 1918, 1930, 1940s, swine 1976, 77, 84, 91, 2007, and we use controls H3 antibodies, antigenically very different to H1s. And then we generate these antibodies in mice, and then we challenge with a lethal dose 50, with a lethal dose of the pandemic H1N1 virus, and then we ask the question, what type of antibodies against what type of H1s are able to protect against lethal disease with pandemic H1N1? And the answer was very clear. So all these vaccinated mice were protected, and they were vaccinated with viruses from the 80s, with 1918 hemagglutinin, from the 40s, and with the homologous strain, or from swine influenza viruses from the 30s or from the 76. But once that we start to use influenza viruses from the 70s, H1s, then there is no protection against lethal disease with H1N1. So the pandemic H1N1 antigenically very similar to viruses that circulated in the 20s, 30s, and 40s, and most likely people that they were born in this period of time that have been exposed to this virus still have antibodies against the pandemic H1N1, and therefore they are spared from disease with pandemic H1N1 virus. Now you can also see that if you look to how much the virus is drifted. So what we have here is the crystal structure of the hemagglutinin, of 1918 hemagglutinin, and here they are included the antigenic sites that are responsible for neutralization by neutralizing antibodies in people that have been vaccinated or infected, predisposed to influenza. And you can see the same thing here from the top. Here, this one monomer, hemagglutinin is a trimer. So here, 3 trimers. In light blue, they are the antigenic sites. In dark blue, they are conserved areas in the hemagglutinin. And the seasonal H1N1s, after this virus has his hemagglutinin being drifted for almost 100 years in humans, they have accumulated enormous amount of changes in the antigenic sites. So antigenically, this hemagglutinin is very different from this one. But the pandemic H1N1, that was the hemagglutinin, was also derived from the 1918, have less changes, and these changes, they are not particularly accumulated in the antigenic sites, and especially this site that appears to be immunodominant is almost identical, no changes in the pandemic H1N1. And that was probably the reason why people that have antibodies against all influenza viruses against this site, they are still able to protect against infection with this new H1N1. So what we learned from these studies is that people carrying antibodies against H1N1 virus that circulated 1918 to 1950, and also that have swine 1976 antibodies, and this could have been done by vaccination. In the United States there was a vaccination because of a swine scare of a virus that they thought it was going to cause a pandemic. A lot of people got vaccinated against this swine 76, and still they have antibodies against this swine 76. They are protected against infection with the pandemic H1N1. And the other thing that we learned is that not only avian acts as reservoirs for new strains of hemagglutinin, but pigs as reservoir for strains that become antigenically frozen. So the H1 went into pigs in 1918, and then it came back from pigs as a pandemic virus in 2009. And there are H3s circulating in pigs. There are new H1s circulating in pigs that have been started circulating from human H3s several years ago. So these ones need to be monitored closely, because at any time you could have the same thing that happened with the pandemic H1N1. We may have a pandemic H3 and 2 coming from pigs into the human population. Now, I show you that antibodies against all influenza viruses H1s will protect against the pandemic H1N1. It's also the same, the opposite, meaning antibodies against the new H1N1 can protect against the 1918. And this is, we just looked to see sera from people that they were vaccinated in collaboration with both Belgi with the pandemic H1N1 vaccine, and we look for the ability to neutralize, here in red, the pandemic H1N1 virus, but also the 1918 virus, and what you can see is there is almost identical ability of this sera to cross-neutralize either the pandemic H1N1 or the 1918. So what this tells us is that the vaccine against the pandemic H1N1 virus is also protecting against the 1918 virus. So if there is any concern about potential bioterrorists making this 1918 virus and put it back into the human population, that will be actually not a good thing for the bioterrorists to do, because we all have now antibodies against this pandemic H1N1, so this 1918 will not be, even the original 1918 will not be as virulent as in 1918 in humans. And not only that, if you take the sera from people that have been vaccinated against 2009 pandemic virus, and then you challenge these mice with 1918 virus, these mice are protected as compared to controls. So vaccination or exposure to new H1N1 virus generates protective immunity not only against the new H1N1 virus, but also against a virus that is 100 years older, the 1918 virus. All right, so I'm going to talk a little bit now about avian flu. Avian flu is due to direct contact between birds and humans, something that has happened since the Greeks started fighting with chickens. You know, that's something that if you are working with an avian influenza virus, that's an example of what you shouldn't do. So you shouldn't use non-protective, you should have protective equipment to prevent infections coming from the animals to you. So these highly pathogenic avian influenza viruses, they are truly highly pathogenic. 1918 estimated number of people that die of 1918 in 1918, 2% of people infected die. H5N1 virus enters a poultry farm, 100% of the chicken die in 2 days. So this is a farm, these are the chickens, that was exposed to a highly pathogenic avian influenza virus. Very different disease from what we know of human influenza. It's a systemic virus, causes neurological disorders, fulminant disease, with viruses present in all parts of the body of these chickens. And how they can become originated, these highly pathogenic viruses. These highly pathogenic viruses come from low pathogenic avian influenza viruses. So this has been always the case. This is how, for example, the H5N1 was originating. There was a low pathogenic H5N1 virus circulating in the wild bird reservoir that somehow got into a farm and then started to circulate in chickens, became adapted to chicken. And at this time it was still low pathogenic. But then a mutation is being selected that increased pathogenicity, and this has to do with, again, what I mentioned before, the cleavage site of the hemagglutinin mutates, and now this virus becomes highly pathogenic. This type of mutations has only been seen in avian influenza viruses, has never been seen in human influenza viruses. And when this mutation arises, then this virus goes from a low pathogenic into a highly pathogenic. How about H7N9? H7N9 is a low pathogenic avian influenza virus, but it's causing now more problems in humans than H5N1. And this is when they were first detected in China in February to October of 2013. There was a large number of human infections in all 137 cases, no human-to-human transmission, in multiple regions in China. And in the second wave that occurs later on, the virus has spread it, and there was a total of 452 now, total cases, with 124 deaths. The case fatality ratio is probably overestimated, so it's probably not as lethal as it is, because it's likely that there is also a lot of asymptomatic infections in people. They don't go to the hospital and they don't get diagnosed. And these are the two waves that happened. The first one was here in February, May. The second one occurred again in 2014. And we are starting now 2015. And we have now 9 new human cases in the Guangdong province over the past 7 days. So it's likely that we're going to get again another peak. Now, these viruses have not been transmitted from human to human, not the H5N1s, but there is a lot of concern whether they will become adapted to transmit from human to human and will start the pandemic. Now we look to H5N1s. They have been circulating more. This is the data from 10 years of circulation of H5N1 in poultry. There is in total 630 infections in China. There is only a total of 45 infections. Now the H7N9 in the same region in China has not been spread to other regions, has caused 10 times more infection in 1 year than H5N1 in 10 years. So H7N9 seems to be, to me, more concerning than H5N1. And the problem is detection of the H7N9. It's a low pathogenic avian influenza virus. The chickens don't die, so they are more difficult to detect. If a chicken dies, then you can look whether there is an H5 there. But if the chicken don't die, usually chickens are not routinely being tested for influenza virus infection. And it appears that most of the infections that have been acquired in humans are associated with contacts in live bird markets between infected chickens and humans, and infected chickens that they were not known to be infected with H7N9. And why this is a problem if a virus becomes transmissible in humans? This is a slide that comes from WHO in 1957 that summarized what happened during the H2 pandemic. And it was first detected in February here in the Southeast Asia, similar to H7N9s, February 1957. By May, it has been spread all over this part of the world, and by August it was all over the world. And this 1957, there was not so much air traffic, there was not so much mobility between people. So once the virus becomes pandemic, influenza virus, this is very efficient for spread. And in terms of vaccines, we are not as efficient to make fast the vaccine to prevent the spread of the virus. This is what happened in the pandemic in the US. In the pandemic, the 2009 pandemic, the peak of infections occurred here in October 2009. The vaccine started to be distributed among people at the time of a peak of infection and at this time there was given to people. So a lot of delay, and this is given the vaccine. Still the vaccine requires something like 3 weeks to induce protective immunity. So the vaccine didn't make any difference in the pandemic 2009. We were very lucky that 2009 was a mild pandemic. If it would have been a severe pandemic, it would have been a complete disaster. And here then I come to my last step of my talk. Can we prevent this problem by making a universal flu vaccine? And the approach that I'm using in collaboration with Peter Palese and Florian Kramer, which are also in Mount Sinai School of Medicine in New York, is to try to induce antibodies against one region in the hemagglutinin that is quite conserved among influenza virus, but that in general does not induce antibodies. And this region is the stalk of the hemagglutinin here in this crystal structure of the hemagglutinin is this area of the stalk. So the globular head of the hemagglutinin is where the antigenic sites are being located. It's quite variable, is immunodominant, and is the one that changes from year to year and also changes dramatically when influenza viruses change of subtype during pandemics. And the antibodies that are being elicited, which are the majority of the antibodies during flu infection or during vaccination against the head, they are very good at neutralizing binding of the hemagglutinin to the receptor. But antibodies against this conserved area of the hemagglutinin stock area, they are also present in humans, but a very low percentage in general. And if one just isolate them and then one test them, they are also able to prevent infection. And they prevent infection by blocking fusion, in general, by blocking fusion of the virus by binding to this conserved area. The problem again is that natural infection or vaccination with influenza vaccines does not induce good level of antibodies against this conserved area of the hemagglutinin. This area over here is immunodominant. Most of the antibodies are against the variable region of the hemagglutinin. Now the conserved area of the hemagglutinin, this one is not completely conserved, but it's quite well conserved between what I call group 1 and group 2. So these are the different hemagglutinins. And the stock is quite well conserved in group 1 of HAs, among all these HAs, and there is a slightly different stock that is well conserved among these different HAs. Antibodies that are directed against the stock 1 in general neutralize stock 1 viruses and not the stock 2, and the opposite. So if one wants a vaccine that is universal, that will be able to protect against all these strains of influenza viruses, one will need to generate antibodies against group 1 stock and against group 2 stock. And how we can do that? Now, what I'm going to tell you is the approach that we are trying to use, and hopefully we will move soon into clinical trials in humans, which is vaccinated repeatedly with influenza virus chimeric hemagglutinin vaccines. And I will tell you what is a chimeric influenza virus hemagglutinin vaccine. And this is in collaboration with a group of Peter Palese and Florian Kramer. So we were able to find that actually you can change the heads of the hemagglutinin without changing the stocks, and this results in a functional hemagglutinin and a functional virus even. If we try to produce the stock alone, the stock that falls apart. It does not, conformationally, you don't get the right conformation. But these chimeric HAs are possible to be produced. So the approach that we are using is we vaccinate with a particular hemagglutinin that contains one stock and a particular head. In this case, it's an H4 head. And then later on, we came out with another protein that has same stock, different head, an H5, another protein, same stock, different head, an H3, and then we look whether we are able to induce antibodies that are able to protect against a completely different hemagglutinin subtype, like day 7 and 9. So the idea is that this head is immunodominant, so this vaccination will induce many antibodies against the head, but some against the stock. But this one now will not boost antibodies against the head because it's a completely new head. Now, during natural exposure to influenza virus or during vaccination, seasonal vaccination, the heads almost doesn't change, so you still induce a lot of antibodies. You are boosting a lot of antibodies against the head. Here we hope that the head is completely naive. You get some antibodies against the head, but now you start to boost the common area of these hemagglutinins, and by the time that you have done this 3 times, then you get a lot of antibodies against the stock that will be able to prevent and neutralize infection with these H7 and 9. So when we do this vaccination in mice, it works. Mice that have been immunized with these different hemagglutinins, but not with an H7 hemagglutininin, the only thing that H7 has in common is the stock, they are protected against death with an H7 and 9, and these are the controls. And this correlates with lower number of virus titers, and we can protect against H10s or H3s, different H3s, even if we use hemagglutinins that they have not H3 head, as far as they have the stock of H3. This is due to antibodies, because if we take the sera from these immunized mice and then we pass it transferally to non-immunized mice and then we challenge them, they become protected. So these are antibodies against the stock. And these provide protection against all these group 2 of viruses that I've shown you, H3s, H7s, etc. Can we protect also against H1s? Against H1s, we need a similar approach by using now a stock from the group 1 viruses. In this case, it's an H9, followed by an H6, followed by an H5 head, same stock, and then we challenge now with different viruses, group 1, and then again we get protection against disease, protection against death. And also we use an H5 or an H6. So what we are getting is full coverage of the total spectrum of influenza virus hemagglutinins, at least in mice, by using immunizations with group 1 and group 2 stocks influenza viruses. Can protection correlates with antibodies against the stock. These are antibodies against the stock of the hemagglutinin, and passive transfer of sera against protect. So both for group 1 and group 2, this seems to be equivalent. Now we use a different animal model that is believed to be more close to what are human infections, which is the ferret model. And we immunize ferrets the same way. We get also protection, at least against replication of the virus, both in nasal washes, in olfactory bulbs, in nasal turbinates, and in lungs. So we think that we have a vaccine now that works in mice, that works in ferrets, and we hope that this vaccine will also work in humans. Now I just want to finish, we're going to finish about what is needed now for actually getting rid or at least managing influenza. One thing that is very important, and we have learned over the years, that we need a better surveillance and eradication of viruses that are in the domestic reservoir. So all the cases that have been of pandemic influenza viruses came to humans. They came from domestic animals that have hemagglutinins that they didn't correspond antigenically to the hemagglutinins that they were circulating in humans at this time. When the pandemic H1N1 came, we had even no idea that this virus existed somewhere. So in order to be able to prevent a pandemic influenza virus, the first is to detect the pandemic virus, surveillance, but also not only detect potential pandemic virus, eradicate them. We cannot afford the possibility that this virus continues still circulating in domestic animals, causing infection to humans. Nothing happened till suddenly something happened and the virus becomes transmissible in humans. We also need new antivirals. I have not talked too much about it, but we have right now only one single antiviral to treat influenza virus infections, which are the neuraminidase inhibitors. And we know that some years the virus can become highly resistant to neuraminidase inhibitors. This is not the case right now, but had happened before and is likely then to happen in the future. So we need new antivirals. And vaccines, as I mentioned, we need broadly neutralizing vaccines. We don't have time to prepare a vaccine that will be able to protect us against a pandemic influenza virus. And we cannot stockpile every potential vaccine against every potential influenza virus, because we don't know which one is going to cause the pandemic. So we need better vaccines that induce broadly neutralizing responses that will be able to prevent not only antigenic drift, but also antigenic shift. And I think also very important that I always mention in my talks is we need to train future investigators. So there is a lot of problems happening lately with respect to research funding, not only here in Spain but also in the United States. This not only impacts already establishing programs of research, but also puts a lot of discouragement into young people that they don't want or they don't have the possibility of becoming trained as academic researchers because they don't have future or because they don't have a job to be able to get to an academic research lab. And 20 years from now, I don't think we will still have, maybe we start to have some vaccine, universal vaccine against flu, but flu still will be with us 20 years from now, and what we may not be with us 20 years from now is a good group of young people now that are actually leading influenza virus research, because mainly some of the approaches that we are using, they are not good, and we need the future people being able to take out and optimize and getting better than we are. And we will not be able to do that unless we train these people. And with that, I finish, and thanks.